Welcome to the Child Mind Institute's 12th annual On the Shoulders of Giants Scientific Symposium, which truly celebrates excellence in research and child and adolescent mental health and pediatric neuroscience, as well as the importance of mentorship. Today, we are honoring the work of Dr. Margaret McCarthy, the winner of the Child Mind Institute's 2022 Sarah Gunn Prize for Research and Mentorship in Child Mental Health. Dr. McCarthy is the James and Carolyn Frankel Dean's Professor, Chair of the Department of Pharmacology, and the Director of the Program in Neuroscience at the University of Maryland School of Medicine. We'll also hear from her protégés, Drs. Jacqueline Schwarz and Dr. Brittany Osborne, illustrating how the new generation stands on the shoulders of giants like Dr. McCarthy. The topic today is the origin of sex differences in the brain and how they impact development, behavior, and psychiatric disorders. This work has the real potential to improve outcomes for our children by enhancing our understanding of the brain. And that connection, science today, for a better care tomorrow, is central to the mission of the Child Mind Institute. For those of you who don't know us, the Child Mind Institute is dedicated to transforming the lives of children and families struggling with mental health and learning disorders by giving them the help they need to thrive. We are the leading independent nonprofit in children's mental health, operating 3 mission areas that truly work together for greater impact: care, education, and science. Our clinicians provide gold standard, evidence-based treatment to thousands of kids annually through our clinical centers and in-school programs. We deliver free resources to millions of families and training to hundreds of educators each year so more kids get the help they need. And our researchers and engineers are uncovering the causes of mental health and learning disorders to develop tomorrow's breakthrough treatments. Our science program at the Child Mind Institute is a global leader in open source innovation and data sharing in neuroscience, brain imaging, wearable behavioral health technologies, and internet-based treatment and assessment. Our tools for discovery are being used around the world, and the work of our engineers and data scientists is changing how researchers do science everywhere. Our Healthy Brain Network study is enrolling 7,500 young people with mental health and learning disorders to uncover the biomarkers of these disorders. And our Autism Center is studying changes in the developing brain in young children with autism. And our newest initiatives are testing clinical interventions and real-world tracking of children's mood, sleep, and activity. In all, our research programs, families receive comprehensive evaluations, recommendations, and treatment referrals. But before we can do any of this, we must transform the culture of science into one that values collaboration. And that's what our presenters today represent so perfectly. Before we begin the presentations, I have a few pieces of business to attend to. First, just before this event, we held a ceremony for our annual Rising Scientists Scholarship winners. Each year, we recognize 5 high school students across the United States who have demonstrated exceptional promise in child mental health and neuroscience research. Hunter College and the Child Mind Institute sponsor a scholarship which gives each winning student $2,000. And this year, we received over 100 highly competitive nominations from over 26 states and from more than 100 schools. And I'd like to take a moment to recognize the winners. I hope you'll join me in appreciating these remarkable young people. Amit Vasantha from Basis Independent Silicon Valley in San Jose, California. Hannah Cho from North Hollywood High School in North Hollywood, California. Lena Ayer from Novi High School in Novi, Michigan. Noemi Duran from Basis DC in Washington, DC. And Ty Gelman from John F. Kennedy High School in Belmore, New York. Second, after each of these presentations, we'll take a few questions from the audience. If you have a question, please type it in the questions window on the Zoom panel, and questions will be reviewed and presented to the speakers. And finally, at 6:30, after a brief break, I hope you'll all rejoin us for what promises to be a remarkable roundtable discussion focused on the implications of Dr. McCarthy's research for mental health outcomes and practical information for parents. Moderator Dr. Kathleen Marikangis, intramural senior investigator of the Genetic Epidemiology Research Branch at the National Institute of Mental Health and also the co-chair of the Child Mind Institute's Scientific Research Council, will be joined by Drs. McCarthy, Schwarz, and Osborne, as well as 2 renowned experts in the field of mental health and child development, Dr. Adriana DiMartino, the research director of the Autism Center at the Child Mind Institute, and Dr. Meng Chuan Lei, clinical scientist and O'Brien Scholar at the Hospital for Sick Children at the University of Toronto. To introduce our first presenter, please welcome my very good friend, learning specialist and philanthropist, Sarah Gund.
Welcome, and thank you for joining us for the 12th annual On the Shoulders of Giants Scientific Symposium. Each year, the recipient of the Sarah Gund Prize for Research and Mentorship in Child Mental Health is chosen based on scientific achievement, commitment to community, and a passion for mentoring the next generation. I'd like to thank the Child Mind Institute's Scientific Research Council for their hard work in selecting a remarkable recipient for 2022. It's an honor that this prize carries my name and that I get to bestow it on an incredibly deserving scientist, Margaret McCarthy. Dr. McCarthy's lab conducted some of the first studies on how sex hormones guide the developing brain to organize differently, and how this leads to differences in physiology and behavior. She believes that by increasing our understanding of how the brain develops differently in males and females, we can find what she calls points of vulnerability for mental health disorders, and also opportunities for intervention. Dr. McCarthy has published over 200 peer-reviewed manuscripts, and her work has been cited over 20,000 times. She's a fellow and a member at large of the American Association for the Advancement of Science, a fellow in the American College of Neuropsychopharmacology, and is the former president of the Organization for the Study of Sex Differences and president-elect of the Society for Behavioral Neuroendocrinology. She was named one of Maryland's top 100 women in 2009 and awarded the University of Maryland's Researcher of the Year and Champion in Excellence 2015 and 17. More recently, Dr. McCarthy's distinguished career was recognized by awarding the James and Carolyn Frankel Dean's Professorship. Ladies and gentlemen, please help me welcome a true leader in neuroscience and mental health, Dr. Margaret McCarthy.
Good evening. Let me start with my sincerest and humblest thanks, both to Dr. Sarah Gund, to Dr. Koplowitz, president of the Child Mind Institute, and Dr. Kathleen Marikangas, the chair of the Scientific Advisory Council, that selected me for this very special award that I feel very proud to be honored by. I'm going to talk to you today about my research in the origins of sex differences in the brain. And you might ask, well, why is it important to understand origins of sex differences in the brain? In fact, you might even say, couldn't that be dangerous? Couldn't that be used for harm? These are important questions to ask, but my motivations is to try to understand the relative risk of neuropsychiatric and neurological disorders which so profoundly shift across the lifespan. So this is my pizza pie version of the relative risk to males and females of various neuropsychological and neurological disorders. So on the right-hand side, we're looking at male bias disorder, and the size of the pizza slice there is the relative amount of the gender bias. So little boys are more likely to be diagnosed with attention deficit disorders, autism disorders, dyslexia, stuttering, any kind of speech apraxia, and as young adults, they're more likely to be diagnosed with early onset schizophrenia. Whereas women on the left-hand side are more likely to suffer from a variety of affective disorders, from feeding disorders to depression, anxiety, compulsion, post-traumatic stress disorder, autoimmune disorders, and late onset schizophrenia. And the important thing about the distinction between boys and women is the timing of onset of the relative risk of these disorders. All of these disorders can happen at any time, but yet they have a strong bias towards the male disorders being childhood in onset, where the female's disorders are largely post-pubertal and adult in onset. So this profound gender bias and shift across the lifespan compels us to try to understand the biological origins of sex differences in the brain. But one of our challenges is that the brain is such a unique organ in that it is actually designed to build itself. So you're born with a perfectly fully functioning heart, liver, lungs, kidney, etc. But your brain is just a buzzing confusion. And that is by design because it wants to build itself in response to the environment in which it finds itself. And that means that if it's in times of hunger, it's going to learn to be lean, etc., that sort of thing. But the problem when it comes to humans is that every one of our experiences are unique, and they are also highly gendered. From the moment you are born, you're going to be treated differently based on whether or not you're a boy or a girl, and that can actually then imbue itself into the biology of the brain. So in order to try to parse out more simply the non-sort of culturally influenced biology, we turn to animal models. And these are our many animal heroes that have helped us over the years in trying to study sex differences in the brain. And my particular model is the laboratory rat, as is that of my protege, Jackie Schwarz, and her protege, Brittany Osborne, who you're going to hear from shortly. And we study rats because they're highly social, fairly smart animals, and they're easy to work with. But we have a problem when it comes to preclinical animal research when we want to study sex differences, and that is that we don't have a long history of including both male and females in what we call preclinical studies. So this is actually was published by Annalise Berry and Irv Zucker. It's a century work of publications in which they surveyed the sex of the animals being studied. And you can see that in the early part of the 19th century, nobody cared about the sex of the animals, so they didn't even bother to report it because they considered it so unimportant. But then in about the late 60s and 70s, you can see there's this marked shift where suddenly most of the studies are being conducted only in male animals, and very, very few studies, less than 10%, are actually using both studies. Well, what does this mean? This means a lot. It means that our fundamental understanding of the brain rests on data generated almost exclusively in male animals. The textbooks and clinical advice, et cetera, all being based on the male brain. It also means the shifting hormonal status of females across the lifespan is not considered. Disorders with gender bias are often mismatched. Most studies of depressive and affective disorders are done in males, despite being more prevalent in females. And for instance, female resilience to autism spectrum disorders is poorly understood due to lack of preclinical representation. And then lastly, what I hope to show you is the explanatory power of comparing males and females, and this is lost when we only look at one sex. So there are four essential questions when it comes to studying sex differences in the brain: the what, the when, the how, and the why. And so the first question everybody asks is, what are the sex differences in the brain? And people always ask me this, and they want me to give them a very clear answer, a nice little laundry list. Well, this is different in males, this is different in females. But the emerging truth is that nearly every neurological endpoint has been found to vary by sex at some time in some brain regions. So this is my cartoon of everything that happens in neuroscience. Start at conception, formation of the neural tube actually varies by whether you're a male or female fetus. Neurogenesis, glial genesis, migration, dendritic growth, connectivity, myelination, synaptogenesis, synaptic pruning, cell differentiation, cell survival, cell death, epigenetic modifications. All of these endpoints have been found to vary at some time in some region in the brain. Not all in the same direction, not on the same way. It's all very region-specific and sort of a mosaic type pattern, so that we have a high variability of phenotypes of male and female brains based on all these different endpoints. Okay, so that's not very gratifying. So when are these sex differences occurring? Well, by and large, they're really established early in development during a sensitive period. So in our rodent animal models, that sensitive period begins embryonically, and it extends out until the first week of life. And this is when all those cellular processes are occurring, and they can be modified along different differential pathways in males versus females. In humans, near as we can tell, based on non-human primate studies and what we know about human brain development, the entire process appears to be prenatal. So they go through a process of brain sexual differentiation before you're even born. Well, how do these sex differences happen? Overwhelmingly, an important part of the sex difference is gonadal steroids. So as you saw during that sensitive period, that sort of red hump there, that is male testosterone levels. A lot of times people think males only make testosterone when it comes to puberty, but actually a male fetus produces copious quantities of testosterone, so much so that a newborn male can be born with the circulating levels of testosterone equal to a 25-year-old adult. But it's only transient, and as far as we know, it's only there for purposes of sex differentiating the brain. What happens in our rodents, as I mentioned, they remain in that sensitive period until after they're born, and this is very fortuitous because it means that we can take a newborn female rat pup and we can actually just inject her with testosterone on the day she's born and 1 day later, and we can literally convert her brain into a, quote, male brain while still in a female body. And then we raise her to adulthood and we can take what would be her normal female, say, sexual receptivity behavior and show that she now will behave as a male if we give her testosterone as an adult. And so that gives us a wonderful tool to look mechanistically at how the brain is developing. But how are the steroids doing that, right? You just told me the steroids. We've known that actually, the secret told, for a good 50 years. But what are the steroids actually doing? And that has been the focus of my research program for the last couple decades. Well, the answer is it's complex, it's multifactorial. There are changes in excitation-inhibition ratios. They enhance growth and survival. They make epigenetic modifications. There are X and Y chromosome genes. And then there's harnessing the immune system. And that's when a neuroscientist says, what? What do you mean harnessing the immune system? The brain is not it's supposed to be immune privileged. It's not supposed to have an immune system. Well, over the course of my attempts to look into the mechanistic basis of steroid-mediated sex differentiation in the brain, I kept making the surprising discovery of roles for immune signaling molecules, inflammatory signaling molecules, and immune cells. So since I can't tell you all that background in this short amount of time, what I want to do is give you one deep dive through one brain region, which I'm going to connect to one behavior. That brain region is the medial amygdala. You might have heard of the amygdala before. It's very important to emotionality, fear, stress responding. But I'm going to talk about a very specific behavior, juvenile social play. So why play? Well, first of all, it occurs only during a restricted developmental period, which is adolescence, meaning post-separation from mother, weaning, prior to puberty, so as a young animal. It is across all species that play, more physical, intense, and frequent in males. Females play, they simply don't play as intensely and as frequently as males. That sex difference, however, is established early, but when it occurs is independent of hormones. So when I study this behavior, I don't have to worry about my animal's estrous cycle or males having testosterone, females having estrogen. Nobody has any steroids. And when young children are playing, they also don't have any steroids on board. And so it has strong face validity with humans and a large range of other species. Adolescent rats love to play. So they will pin each other, they'll box with each other, they'll pounce on each other, and they'll just plain wrestle around. And so how we study this is we go in the afternoon and we give the animals a play date. And we set them up in this neutral arena up in our animal facility, and they meet their playmate for about 10 minutes. And what you're seeing is there's soft bedding there that they're on. And you see on the top we have two males who are having a great time. I have not sped up this video. This is real time. This is how fast they're moving. And on the bottom you see two females. They know each other are there. They're very social. They're glad that they have their friend to hang out with for a period of time, but they simply don't show the same robust amount of playing around. You might notice the black marks on one animal's head. That's how we tell the two animals apart, because we're trying to score their behavior. And that's what we do, is we quantify how frequently they play and the like. So as I mentioned, that this is a sexually differentiated behavior, so what we can do is we can take that newborn female, give her testosterone, and convert her brain to a male in terms of the medial amygdala, and just promote her playfulness. And then we want to know what is the mechanism. So about 12 years ago now, we published a paper in which we had identified the mechanism, which was that it was a sex difference in cell proliferation in the developing rat amygdala, and that this was the basis for the sex difference in social play. And what you're seeing is that females, you see the red bar in the histogram there, is females' newborn cells versus the male blues. And those two panels with the little dots, those are the newborn cells that we visualized with a DNA analog called BRDU. We published this in a very prestigious journal, the Proceedings of the National Academy of Sciences. We were quite confident that we had done all of the appropriate control studies to show that it was indeed cell proliferation, but we were wrong. And it's always good to discover your own mistakes rather than having somebody else do so. So how was it that we were wrong? Well, we hadn't considered all of the potential variables. So the brain has dozens of distinct cell types. We're all used to just thinking about neurons. We've now gotten used to thinking about astrocytes, also called glia, the glue of the brain. But what's only recently been getting a lot of attention is a third cell type called microglia. You can see them down there in the right-hand corner. They're very small cells. That's why they're called micro, but they're actually 10% of your brain. And they look like astrocytes, which is why they're called glia, but they are nothing of the sort. They are actually macrophages, modified macrophages in the brain. I just would skip over that. And this is what they look like in the brain. This is a cross-section through the brain. This is what we call coronal, that the white space in the middle there, that's the third ventricle of the brain. And all those little brown spidery things that you're seeing, those are the brain's immune system. They are tiled throughout the entire brain. They're covering the entire brain. The little green ovals there, that's another brain region called the sexually dimorphic nucleus that we also study. So I just want you to get the sense that these cells are embedded in the brain, surrounded by other neurons and astrocytes. And what they usually look like is like this, with these long processes that we're now visualizing it here with immunofluorescence. And they're actually reaching out and touching all of the cells around them and sort of checking on them. Every microglia has a stable of cells that it checks on a regular basis, until occasionally as you see them doing this. And what you're looking at, those big cup-like things, which is what the graduate student called them when they saw them, those are phagocytic cups. As you can see, this one particular microglia is a very, very phagocytic cell with up to 3 phagocytic cups from just that one cell. So, and this is again what they look like. This is real live imaging with using immunohistochemistry to show how they're everywhere in the brain. And we just didn't even, we sort of ignored them for many, many years. So Jonathan Van Rissen in the lab asked a simple question: is there a sex difference in the microgliophagocytosis, those phagocytic cups, during the sensitive period? So he just stained to visualize the amygdala, and he just went in and counted them across that entire sensitive period. That P0 is the day of birth. P8 means they're 1 week old. And what you can see is that the blue is the males. I should have labeled that, sorry. It's always obvious. Blue is males, red is females. And those male microglia during the first 4 days are consistently more phagocytic than the female. And then they both stop being phagocytic by the end of the time. So it's only happening in a very short period of time, which if we had not looked at that time, we wouldn't have seen it. And if we had not compared males and females, we wouldn't have known that it had any kind of important regulatory role. So the big question is, what are they eating? Well, based on the size of those big phagocytic cups, we were pretty sure they were eating other cells. But since we knew the females had more newborn cells, we wanted to know, is that preferentially what they're eating? This is a more sophisticated type of imaging called IMARIS. This is one microglia that we've sort of pulled out from the surround. You can see what gorgeous cells they are. They almost look a little Halloweenish, a little ghoulish. And we can tell a newborn cell by that red stain. And you can see this microglia has reached its arm out and engulfed a newborn cell, which it's holding in its clutches. You can also see that big red oval is the cell body of the microglia, showing it is also newborn. So both of these cells were born within the previous 48 hours, which is a sure sign that this is an immune cell, not a neuron or an astrocyte. Here's another image of a microglia eating a newborn cell. I call this my catching the home run ball over the wall microglia. You can see it sort of reaching over. We see this all the time. They seem to be reaching out and saying, you're the one I want to eat, you look yummy, and they grab it and gobble it up. What I'm going to focus in on here is the inside of the phagocytic cup. That blue circle you're seeing, that's the nucleus of the cell. It's fully intact, fully healthy, which tells us this was a living cell when it was engulfed. If it was a dying cell, that nucleus would be broken up into bits, because that's the first thing that happens when a cell is dying. And in fact, 60% of those cups contained newborn cells. So these microglia are selectively picking out brand new cells. We went on to confirm that those cells were indeed alive. This is a process that's only been named in the last 10 years, in which we find out that there is a phagocytosis that's called phagoptosis, because it refers to apoptosis combined with phagocytosis, in which the microglia will engulf a living cell and literally kill it and digest it. And this is going on in the brain. It's just kind of stunning to think of. It still shocks me every day to think about it. It's pretty, the brain never ceases to surprise. What were those newborn cells going to become? They were actually going to become astrocytes, not neurons. And so what happens is, in the male, they eat more of these astrocyte progenitors because their microglia are more phagocytic. And that leaves the female with more newborn cells that go on to live a long and happy life, so that she winds up with more astrocytes just in this brain region controlling adolescent play. So the long-term consequences are that she has more astrocytes, and that dampens neuronal excitability related to play, so she plays less, whereas the males have fewer astrocytes, allowing those neurons to be highly excitable, and results in more play. This is a very exquisitely, tightly, evolutionarily controlled behavior. So it must be important, right? And that is the big question. does play with others really matter? This is actually, seems like obviously it must be, but this has been debated by psychologists, psychiatrists, teachers, and parents, and we still don't have real definitive evidence. One of the reasons that we don't is because in our animal models, the only way we've been able to study it is to completely socially deprive animals. That's about the worst thing you can do to an adolescent, is completely isolate them. It's very, very stressful. So Ashley Marquardt in the lab developed this caging apparatus where she put in this perforated barrier, and she can put two adolescents in there, and they can see, smell, hear, and a little bit touch each other. They actually nestle against each other right across the barrier, but they can't play. They can't do rough and tumble. And this, I will say, is the meanest thing we do to our rats. We don't addict them to drugs. We don't foot shock them or anything. We just block them from playing during the entire adolescent period, and then raise them to adulthood and say, how is your sociosexual behavior? Is it normal? Did not being able to play but still having social contact influence you? We first looked at the males and their adult sex behavior, and when you quantify male sex behavior, you measure how frequently they mount a sexually receptive female, and we found that these males were in fact fairly severely impaired, whether or not they were housed in this barrier housing or completely isolated. But even more impressively is that they were significantly less likely to ejaculate on a timed trial. And if you want to talk about evolutionary significance, that's a real penalty when it comes to your reproductive fitness. We also looked at their aggressive behavior. We did a resident intruder paradigm where we put a stranger into their cage. Everybody often talks about play as play fighting and the idea that it makes you a better fighter when you get older, but actually these guys not being allowed to play were more aggressive than they should have been, sort of adversely aggressive, which we believe is because they are not properly reading the signals of the intruder and instead attacking them excessively. So play matters. But what about females? We didn't see any effect on their sex behavior. We didn't measure their aggression because they're not very aggressive. But we did turn to another test of what we call empathy, where we take two cage mates and we put one animal in a restrainer there. And then that funnel there, we fill that with ice, and we actually drip cold water on the animal in the restrainer. They absolutely hate it. And they start calling out to their cage mate, get me out of here, and there's a little trip door that they have to learn how to open up to release their cage mate, and this is considered a test of social empathy. The females and the males who were in the barrier housing, not allowed to play, they were incapable of showing this kind of empathy. I shouldn't say incapable. They took a lot longer to release their cage mate from the restrainer and to understand that the other animal was in distress. So we conclude that microglia, which are immune cells, control the number of astrocytes, also called glia, in a specific brain region, the amygdala, to regulate adolescent social behavior, play behavior. But engaging in play promotes further brain development required for appropriate adult sociosexual behaviors. But we also think the purpose of play is different in males and females, and that is part of our ongoing research right now. And last 2 seconds, what does any of this mean for human brain development? Well, as I told you in the beginning, we have this enormous gender bias across the lifespan, and that being male is the leading biological predictor of your relative risk for a developmental neuropsychiatric disorder. What I didn't bring up is that early life inflammation is the leading environmental predictor of your relative risk of a developmental neuropsychiatric disorder. And so we wonder, are these two somehow converging to make a greater risk for males that just pushes them closer to a vulnerability threshold in a very simplistic sense? Jacqueline Schwarz, who's going to speak next, is going to speak much more about early life inflammation and how it can increase vulnerability to later cognitive impairment. Thanks to the funding agencies, my many wonderful postdocs, graduate students, and technicians. This is a photo from the before times, a Halloween many years ago, in which we all dressed up as our favorite immune cells, as you can see here. And then here's everybody from my laboratory, and highlighting there Jacqueline Schwarz. And thank you very much for your attention, and I'm happy to answer any questions. I have a question here that says, what about those newborn cells tells the microglia to turn around and eat them right away? That's an excellent question. I didn't go into detail on what is called the complement system. So every cell is coded with a combination of what we call eat me signals and don't eat me signals. And we found that the newborn cells in general are coded with more eat me signals, which come from the complement system, which has to do with the body fighting off bacterial infection. It's kind of, again, a rather terrible name because it doesn't really give you much information. But this is the same sort of signals that has been identified recently in synaptic pruning and the like. If we block the ability of the microglia to see the complement signal, we can block the phagocytosis, and we can actually allow the cells to survive in the females, so that then they have the same number of astrocytes as males, and then they start to play just like males. And that's how we know we can causally connect this cell population control to the behavior. Okay, I'm not seeing anything. Let's see. How long does it take the microglia to shape a brain structure during development? Another really excellent question. So in the circumstance of the medial amygdala, which we just saw, this all happens so quickly. It's over a matter of no longer than a week. We don't know what's going on prenatally, but after about 5 days postnatally, it's all done. So a very, very narrow critical window, sensitive period. And the brain is probably full of similar epochs. We do know of one other that is actually happening about a whole week later in another brain region where the microglia are actually engulfing mature neurons, more so in this case in females than in males, and so that more neurons are surviving in the male in that particular brain region. And so this is one of the challenges that we have, is we have to look at every single brain region independently, because nature didn't do us the favor of generating a unified field theory of sexual differentiation. Instead, every region has its own little set of rules, which in the end makes sense because it greatly increases variability across individuals. But the real important point is that these sensitive periods are very short, probably many, many more of them than we know of, and hidden. Okay, what happens if a sickness diverted the attention of the immune cells from performing these functions during development? Yes, that's absolutely right. Now, we have, it would derail it, one would assume. Now, you might have noticed if you were paying close attention when I put up that paper that we're studying, that this whole process we found is actually mediated by endocannabinoids. Endocannabinoids are the brain's own marijuana, right? And so we have also done studies where we have actually tried to mimic the use of cannabis during pregnancy, and we have found that that actually disrupts this whole process because it interferes with the regular signaling process. In other studies, we have found that if we make animals sick very early in development, it disrupts their play. We haven't tied it exactly to this process, but it probably disrupts this and other regulatory processes mediated by this endogenous immune system, depending on when the illness happens. So it's all about a convergence of the time of the illness and what's going on in the brain at that particular role. What is the role of culture in playing behaviors? Well, certainly in humans, it's huge, right? Let's use our inside voices, girls. Stop crying, young man, or I'll give you something to cry about. Oh, girls don't, get their knees dirty. It's probably enormous. And I would not want to say that there has not been cultural influences in humans. It does seem, though, that, every species that we look at, with one exception, which is the spotted hyena, in which females play more than males. If you know anything about spotted hyenas, you know why that's an interesting exception. And if you're curious, I suggest you go Google it. That humans, again, follow the exact same pattern. And I should also point out there's tremendous individual variability both in our rats and in humans. We have some females who play at a very high rate, and we have some males who are not very playful at all. And in fact, Ashley Marquardt has been looking at the transcriptome of the medial amygdala in those animals and finding different patterns. So I'm talking about averages across groups, but there certainly is a lot of individual variability. And in humans, that's going to also be hugely impacted about your parental environment, your school environment. Did you go to an all-girls school, all-boys school? It's so many things. How many siblings do you have? The gender of your siblings has a huge influence on your behavior preferences early in life. Okay, last question. Can you rehabilitate the shift in empathy in one sex versus the other? Well, that's a fascinating question. We're just really getting going on these studies, and that is something we will certainly be interested to look at. Rehabilitation in general is a very interesting idea. Whether or not these changes are epigenetic and they can be unwound will be something that we would really like to look forward to talking about later on today in the workshop session. So thank you very much for your questions, and I would now have the great honor to introduce my protege, Dr. Jacqueline Schwarz. So Jackie received her PhD with me at the University of Maryland Program in Neuroscience in the School of Medicine. She then went on to do a postdoctoral training in the laboratory of Dr. Stacy Bilbo at Duke University before taking a faculty position at the University of Delaware in 2013, where she has risen through the ranks and is now a tenured associate professor.
Thank you, Peg. I am so honored to be a part of this amazing award that my former PhD advisor has been awarded. And I can't say enough about the impact that she's had in research, but also as a mentor to me and so many others that I know in our field of developmental neuroscience. So today I'm hoping to talk to you about my work, which has been exploring the effects of early life immune activation and how that might perturb the development of learning. We're trying to understand the factors that increase the risk of learning disorders in children. And first I have to convince you that the activation of the immune system affects your brain and behavior. And this shouldn't be tough. So let's say you're sick, you have COVID, you get some kind of bacteria, some virus, you're sick, you get activation of the peripheral immune system. This activation of the peripheral immune system is meant to attack the virus, but what it does is it triggers the production of cytokines in the brain and a whole host of sickness behaviors that we are all too familiar with. Reduced appetite, increased sleep, decreased activity, altered cognitive function, right? You're trying to read something and you just can't think of it. And reduced social interactions, right? Nobody wants to hang out with each other. You just want to go to bed. And this is adaptive. It's a conserved behavioral response to immune activation. It's conserved across many species. Ultimately, a central change in the organism's priorities to cope with the infection. Go home, get better, we'll see you tomorrow, right? The problem is that immune activation can be adaptive, like I just described, in order to get through an infection, or it can be pathological. So rather than being sick, it is very possible that in the absence of any overt sickness, that there is some sort of brain immune dysregulation. So elevated levels of cytokines that are produced by microglia, those immune cells that Peg just described. And what that does is it induces a whole number of dysregulations in immune system, as well as a number of behavioral symptoms for various mental health disorders. So these symptoms, interestingly, for many child mental health disorders or adult mental health disorders actually mirror a lot of the sickness behaviors that I just described. So in this case, we see chronic metabolic disturbances, chronic sleep disturbances, fatigue, lack of energy, altered social interactions, and increased withdrawal or depression. And what I'm most interested in is decreased cognitive function. That this dysregulated immune function is known to affect learning disabilities, dementia, and at a worst case scenario, delirium. Moreover, microglial activation has been associated with autism, schizophrenia, cerebral palsy, pervasive developmental disorders, depression, addiction, and neurodegenerative disorders. They're falling off the list, but equally important. And microglial activation in postmortem tissues point to the fact that the immune system is dysregulated in these disorders. So our research question has revolved around this idea, which is that immune activation or dysregulation early in life is linked to the etiology of neurodevelopmental disorders. So we have a lot of evidence to suggest that early life exposure to infections, say prenatally, and associated inflammatory responses have been implicated in the etiology of autism, cerebral palsy, and schizophrenia. Individuals with autism, schizophrenia, cerebral palsy have altered immune function, increased antibody production, some of which is targeted towards brain cells, altered T, B, and natural killer cell immune responses, elevated cytokine production in the bloodstream, and within families with autism and these disorders, increased incidence of allergies and autoimmune diseases. And then, of course, early life immune activation or dysregulation has just been associated with generalized pervasive developmental disorders. And when we look closely at some of the mechanisms, we're starting to understand that the network analysis of genes in both normal and autistic brains reveals a change in both synaptic and immune genes, suggesting that the immune genes are corresponding to synaptic function that likely results in these mental health disorders. So my lab in particular is interested in understanding how immune activation during brain development results in the emergence of learning deficits or disorders. So how do we do that in a rat? Well, they're actually extremely smart. They're so smart. And what we know is that rats, just like humans, begin to be capable of doing very sophisticated cognitive tasks at specific ages. So the data you're seeing here, it uses a hippocampal-dependent context learning task that we can do around the time of weaning. Very early on, when they are good at this task, they start to show freezing, right? They were in a context, they got shocked, they didn't like it, they'll remember next time, and they freeze. And we start to see that increase in freezing in the rats as soon as they turn to be about 3 weeks of age, and then 3 days later it gets even better. So that tells us that during this time, about 3 weeks of age and 3 days later, that there's some ongoing changes in the brain that allow for these juvenile rats to begin to learn context-dependent environmental stimuli. So our hypothesis is that immune activation during this unique period of hippocampal development will produce deficits in the emergence of learning. To that end, we've had quite an interesting ride. So what we do here is we provide the rats, these are juveniles, they're 3 weeks of age on postnatal day 21, a very low dose of what's called lipopolysaccharide. It triggers the immune system in a very controlled way. It's part of a bacterial cell wall. Then we wait 3 days and we test them in our context-dependent learning task. So they go into the context, they explore around, they're walking around. Next day, bring them back, give them an immediate shock that's aversive, but makes them say, where was I? And then day 3, they come back and we see an increase in freezing because they know, I got shocked here. But interestingly, the rats that had LPS, and we're talking 5 days prior, show deficits in learning. So a main effect here of the immune activation that disrupt the emergence of the learning. Interestingly, we can shift that immune challenge. So in this case, we have tested it when learning has already emerged. So on postnatal day 24, rats know how to do this task, but let's give them an immune challenge, wait 3 days, and put them through the paradigm. Show them the context, give them a quick shock, bring them back 3 days later, and there are no learning deficits. So what's that allowed to do is it's allowed us to say that there's something unique about that period of development when the circuits were probably wiring themselves, that the immune system dysregulated that process and thus resulted in learning deficits days later. Because I learned from BEST, I learned from PEG, I also study males and females. And in this case, we use the same paradigm and administered the immune activation to both males and females in these juveniles, and we found a similar effect. Both males and females were affected and showed learning deficits at postnatal day 26 when they were tested. Interestingly, though, I will show you here, I'm highlighting that the Cohen's D, the effect size in males was greater than it was in females. So what is happening? Well, my graduate student, Brittany Osborne, who you'll hear from next, did a great extensive look at what's going on in the brain. And what she found was that immediately after that acute immune challenge, we see a robust inflammatory response that's associated with immune activation at 3 weeks of age. So we see an increase in cytokines, these inflammatory ones, IL-6, IL-1 beta, TNF alpha, all of that occurs within 24 hours and it's resolved within 24 hours. Okay, so the animals aren't overtly sick by the time we're testing them for learning. Great, but what happened in the meantime? It turns out that all of those acute immune changes then initiated a change in the signaling between neurons across synapses and microglia that only begin at 24 hours after the initial immune challenge. And what we're seeing is a whole host of molecules, including a decrease in brain-derived neurotrophic factor, not good for learning, a decrease in CX3CL1, that's a fractalkine, that's a molecule coming from neurons that's telling microglia something's not right. And in turn, we see an increase in phagocytosis from microglia, indicating that they are busy trying to rewire what's going on or adapt to what just happened. And so now we're taking that to the bigger picture. What you're seeing here is diffusion tensor imaging of the rat brain. And what's so wonderful is that we really know the circuits that are involved in this type of learning, contextual learning. And these circuits begin to emerge during this time. So we're looking at a rat that's 16 days old, 20 days old, and 24 days old. And look at the number of connections between, within, and outside of the hippocampus. It's a brain region that is essential to this behavioral task. But now we can take MRI in a live rat and say, what do these circuits look like in the presence of the immune challenge that happened, you know, on day 21? How does that look 3 days later when it should be developing appropriately? So we can get a sense of how the dysregulation of the immune system and the associated cytokine production ultimately integrates into the networks that are necessary for these learning, and then how we end up with these poor pups that have learning deficits or delays in learning. So it's nice as an approach because we can do the same thing with children and understand a little bit about the circuits that get rewired in the presence or shortly after an immune challenge. So I'd just like to thank, of course, my PhD advisor, the winner of the Sarah Gund Award, Dr. Peg McCarthy. So I just want to thank, of course, my PhD advisor and my friend and my mentor who trained me to understand the sensitive periods of brain development and how sex influences that. And then I also want to thank our next speaker, which is Dr. Brittany Osborne, who did a lot of this work here I presented today. And so I look forward to taking some questions before we hear from my former PhD student, Brittany Osborne. So a question here I have is, when there are so many effects of inflammation on such different disorders, how do we get to specifics? Oh, yes, the scientific officers love this question. Because in science, we want to point to one thing, right? It's very much easier if we can manipulate one molecule, one immune molecule, one type of cell. But we are working with an immune system that coordinates across a number of different signaling molecules to mediate an immune response. So it's a really great question. What I think we're got to get down to is understanding variations in the timing, the duration, the intensity of the immune response. Not every immune challenge produces the same immune response, right? So a virus is detected by the immune system through a different set of innate immune receptors. Bacteria is activating a whole new set of innate immune receptors, and those tend to trigger different downstream signaling pathways. That said, there's still overlap in those molecules. And so I think we can learn a lot about which disorders, neurodevelopmental disorders, may be associated with similar or quite different patterns of immune activation and cytokine production. And hopefully that'll get us somewhere in understanding how we can target therapeutics. But I think the therapeutics are going to have to come from, somewhere downstream of what the immune molecules are actually then doing to the neural circuits, and intervene there, maybe get those microglia to rewire circuits outside their original period of brain development. Another question we have here, does birth by C-section result in lower immune protection for the newborns and thus make them more susceptible to immune activation? All right, so I have 2 points to this question. One is that the birth process itself, not C-section, but the vaginal birth process itself is a very inflammatory event. And it appears that that inflammatory event is an essential component to what the fetus experiences at the time of parturition. I'm going to use the word parturition, but at the time of being born. The second facet of that is that is the microbiome to which the baby is exposed to during the birth process. If it's a C-section, they're not necessarily colonized with all of the microbes that might be colonized in a baby who was passed through a vaginal birth. And so there are two different things to consider in how that influences the immune response in a newborn. That said, I feel pretty confident that newborns, even those that have been delivered through C-section, catch up quite quickly from some data that I've seen in their microbiome to other children that maybe were not C-section. But lots to think about when we're talking about an immune system that's extremely immature within the first few months after birth. Last question here, not last question, but how can you translate this research from rats to humans? So that is a great question. What are the challenges? So I think one of the challenges, obviously, here is that the repertoire of learning in rats is much more limited than what we know in humans. Humans learn a lot more things. That said, rats are very smart. One of the limitations that we have when we're caring about the development of the brain is that the development of the brain occurs during a very short period of time in a rat, and it occurs in a much more prolonged fashion in humans. And so the manipulations that we can do and test in rats have to occur during a very short period of time. That's why I love that test that I used here. It's because it's done in 3 days. Right? It doesn't require 2 weeks worth of training. By that point, the animal would be, it's a very sophisticated task, but the animal would already be an adolescent, right? So we are limited in that kind of timing of development. But I also think that what we benefit from is that the circuits that underlie a lot of these very simple and important behaviors in rats are really well characterized. And that's the best part, is we can take those circuits, we know which ones they are, we can examine how they're disrupted and how they're incorporating some of the immune molecules so that, yeah, we can hopefully answer better questions in a short period of time about how an immune challenge actually impacts the development of a circuit during a pretty rapid period of time. So I hope that helps answer that question. All right, thank you for your questions. I would like to now introduce my protege, my first graduate student, Dr. Brittany Osborne. She graduated from my lab in 2019, not that long ago, and we recently just got a lot of this work published here. An amazing scientist who has now taken on a research scientist position at the Children's Hospital of Philadelphia, looking at immune-based therapies for cancers in the brain. And so, of course, I'm similarly proud of all that she's done, and I look forward to hearing her work today.
Hi. Thank you so much for having me today. I am truly honored to be here and be a part of this event at the Child Mind Institute, honoring Dr. Margaret McCarthy receiving the Sarah Gund Award. So I will get right into my talk today. So I just wanted to begin today by posing the question, why is it important to understand adolescence as a vulnerable period to stress? So this is because what happens in childhood doesn't always stay in childhood. So we know that adults who endure early life stress or traumatic experiences are at an increased risk for developing mental health and substance abuse disorders compared to adults that have not experienced these. So adolescence is a time of tremendous social, emotional, and cognitive development. Adolescents are also exposed to a number of daily stressors that can come in the form of academic pressure, peer and social pressure, as well as bullying. And we know that there are sex differences in some of these experiences. So, for example, boys are more likely to experience physical forms of bullying, whereas girls are more likely to experience social exclusion or emotional bullying. Adolescents are also at a greater risk for more severe traumatic experiences, so things such as abuse and neglect. And in these cases, we see sex differences as well. So boys are more often exposed to physical forms of abuse, while females or girls are more likely to experience sexual abuse. And importantly, the more risk factors adolescents are exposed to, the greater potential impact on their mental health. And so I just wanted to show a little bit of data here, looking at one mental health disorder we know is related to stress in adolescents. So this is showing episodes of major depressive disorder in adolescents in 2020. This is data from the National Institute of Health website. So you can see overall, adolescents are showing around 17% prevalence of this, and that this increases as age increases. Importantly, we also see that females or girls during this time are showing a little over twice as much prevalence of this as males. And this sex difference actually persists across the lifespan. And so what I'm showing here is a table of stress-related mental health disorders. And what I have highlighted in yellow are the mental health mood disorders that are more prevalent in females. And then in blue, I'm showing the alcohol abuse and drug abuse disorders are more prevalent in males. So we know that biological sex is just one factor that confers risk or resilience to the detrimental effects of stress in adolescence. And so in the lab, we study this in rodents, and the most well-studied major player in regulating the stress response in humans and rodents is the hypothalamic pituitary adrenal axis. And so this axis starts in the brain, and in response to an environmental stressor, releases a couple of different important neurohormones into the periphery, and this ultimately results in the release of cortisol in humans. This is also known as corticosterone in rodents. And so this is released into the periphery. And then importantly, this circuit is also self-regulatory under normal healthy conditions. So when you have an environmental stressor that activates this system, you have not only cortisol that's released into the periphery, but some of this cortisol actually feeds back into the brain. And this is known as a negative feedback loop, because this is actually signaling the brain to shut down this stress response. And what we have found when we look at male and female rats, as adults, females actually have a reduced negative feedback to shut off the stress circuit following a stressful event compared to males. And this data is actually reflected when we look at adolescent girls aged 9 to 14, who I'm showing here on the graph with the two asterisks display a higher HPA axis response to this Trier social stress test. So this is a test that involves public speaking as well as 5 minutes of mental math. And so children are actually brought into a laboratory setting and then administered this task. And so you can see those two data points there in black showing that girls are actually displaying a much higher stress response than males. So importantly, while the HPA axis may be the main stress response regulating system, it doesn't work in isolation. So there are a number of other factors, internal and external, that control or regulate the HPA axis. One of those that I've studied in my postdoctoral work is the neurotransmitter orexin. And so this diagram here of the rat brain, I just want to point out in the bottom there, the orexin neuron population is almost exclusively found in the lateral hypothalamus. And all of these arrows are pointing to other brain regions that we know orexin has its effects on. And so you can see that one of the main regions that it affects is the stress HPA axis. It's also involved in a number of other important systems, including arousal and sleep, emotional memory, reward and motivation. And we know these are all key components that can be affected by stress. And they're also affected in a lot of these mental health disorders that we talk about that are related to stress. Previous data from my postdoctoral lab showed that if you give an orexin antagonist, which is going to reduce the amount of orexin available, you can actually reduce the amount of corticosterone available in adult male and female rats. So the first question that I wanted to ask when I started my postdoc was, are there any sex differences in the orexin cell population in adult males and females? And so I looked in the lateral hypothalamus and just counted the number of orexin cells. And what I found was that females actually have more orexin neurons compared to males. And that's represented here by the bright green cell bodies. Those are orexin cells. And then I just have this quantified here in the graph as well, showing that females have more orexin neurons than males. And so importantly, though, I was very interested in this adolescent period of brain development. And what does this orexin system look like in the developing adolescent brain? And so this graph is showing across age here. So postnatal day 21 was the earliest that we looked. So this is around weaning, when rats are able to be independent, but they're still very young. And then postnatal day 30, that second time period, this is considered sort of a prime adolescent period in rat development. And then P45 is right around puberty for rats. And so what I'm showing here is that at that postnatal day 45 period, you see a significant decrease in the number of orexin cells compared to these other two earlier developmental time periods. And this is more closely at P45 showing what we found in adulthood. So I was, of course, very interested in this postnatal day 30 time point. And so the next question I wanted to ask was whether or not there were any biological sex differences in the adolescent orexin system that may inform sex differences and vulnerability to stress. And so to do this, I wanted to look at orexin cells and their interactions with microglial cells. And if you've been here for Dr. McCarthy's talk and Dr. Schwarz's talk, you know a little bit about microglia and how important they are for regulating and communicating with the neuronal cell populations that surround them. And so I particularly wanted to look at a measure of distance. So how closely in proximity are these cells interacting? Because this gives us an approximate measure of whether or not these orexin cells and microglial cells may be communicating in a way that's important for understanding functional consequences for the cell population that microglia are interacting with. And what I found at that postnatal day 30, that adolescent time point in rats, is that females actually have more orexin neurons that are within close proximity to a microglial cell, suggesting that these microglial cells are doing something to regulate orexin cell activity in adolescent females that they're not doing in males. So finally, I just want to conclude by saying a couple key points to keep in mind from this talk is that adolescents face a variety of stressors that can impact their mental health and that these can persist across the lifespan. Biological sex is just one factor that we know has been shown to influence the stress response in adolescence. Orexin regulates the stress response, and it may be an important target for understanding sex differences in the adolescent stress response. The immune system, mainly here we're talking about the resident immune cells of the brain microglia, may be important regulators of the sex differences that we see in the orexin system during adolescent brain development specifically. And just a bigger takeaway message, the ultimate goal of all of our basic science research is really to be able to translate these findings to inform clinical treatment and prevention of stress-related mental health disorders in humans. So I'd like to end by just thanking, of course, our funding agencies, as well as all the talented lab members and from my postdoctoral lab and my postdoctoral mentor, Seema Bhatnagar, as well as my PhD advisor, Dr. Jackie Schwarz, who has really had a tremendous impact on my career as a scientist and who's been a phenomenal mentor and learned so much from Dr. McCarthy that I'm just so thankful to be a part of their mentorship across time. So I will take any questions that anyone has. So the first question I have is, why would orexin cells be so vulnerable to stress? That's an excellent question. And I'm not sure I have a concrete answer as to why orexin cells specifically. I think more broadly speaking, if we think about a lot of what Dr. McCarthy spoke about, which is that generally speaking, the brain is part of its environment. And so there's a feed forward system, but there's also a feedback system. And so the brain is wired essentially to adapt to the environment, whether that environment is good or bad. And so I think the orexin system is not perhaps unique in its vulnerability to stress. I think it's just one component of many systems within the brain that would be vulnerable to stress. I think because we know that for whatever reason it is uniquely positioned to regulate the stress response is why it happens to obviously have such a great effect or a profound effect on stress regulation. But I don't think it's any more particularly vulnerable than potentially other systems. So the second question, is the reduced negative feedback in the stress system of females programmed during development or due to cultural influences? That's a really excellent question. And so I don't think that's something that we can fully model in our animal models. If we think about the human data, I can't point to any specific data right now, but I would have to assume that it's not only biological, but there are also, without a doubt, cultural influences on this. As Dr. McCarthy had mentioned, from the time you're born, you're treated differently based on your assumed gender. And these things are encoded into our brain development. So, yeah. Okay, the next question. Am I correct in assuming there's more orexin in the medial than lateral sections of the brain based on the graph? Yes, so I did not show this analysis, but so there are two subdivisions of the lateral hypothalamus. There's the medial portion of it and the lateral portion of it. And I do have data that support the idea that there are differences in the number of orexin cells within those two subdivisions. Like I said, did not talk about that today, but that's a really important finding that could have implications for exactly what circuits and what behaviors these cells are regulating. Okay, next question. Do you think we can utilize orexin as a way to reset the amygdala following trauma or stress? And are there any animal studies that have done this? So, to my knowledge, I do not know of any animal studies that have addressed this, but this is a very interesting question. Whether or not we can use orexin to reset amygdala function, I think it's hypothetically plausible. And one of the ways in which I think that is plausible in a more general sense that could affect the orexin system is something that I think about a lot, which is enrichment. So, you know, you can't always, what we would love to do is be able to prevent a lot of these changes. But clearly, especially in human populations, we cannot always do this. And so I think learning more about how different therapeutic strategies or enrichment strategies may be able to affect the orexin population to then feed forward into that amygdala circuit where we do know that they have projections and affect cell function would be an extremely interesting route to pursue, whether or not we can sort of mitigate the effects of the orexin system dysfunction following stress. Okay, that's all the time we have. So I just want to thank everybody for your questions, and I will now turn it back to Dr. Koplowitz.
Thank you, Brittany, and thank you, Jackie and Peg. What a fascinating group of presentations. We will now take a very brief intermission. We hope you'll join us back here at 6:30 for our panel on the impact of sex on the developing brain.